Hi, I'm Alex Popham. I'm the 4-H agent here in Breckinridge County. I'm part of the University of Kentucky's College of Ag, Food and Environment's Cooperative Extension Service. So today, this first video is going to kick off several weeks of 4-H set activities or science, engineering, technology activities that you can do at home or join me and learn about. The activity today is part of our 4-H Biotechnology curriculum, so definitely check that out. I thought it would be really fun if we kicked off this series learning about DNA. So we'll go ahead and get started. If you look at this model that I have of an animal cell, just like a human cell, one of the most striking features is the nucleus. This is where all the DNA is, and the DNA are the instructions that help the cell function and take care of all of its processes. So the DNA is very important. And just a cool fact about DNA, there are 46 DNA molecules in a single human cell. We call those chromosomes. And if you were to stretch out, like uncoil and stretch out that DNA in a single cell would be about 6 feet long. So you can think about how long that code would be for the entire human body. It's pretty exciting and fascinating to think about. So we'll go ahead and get started with our craft. If you look at the DNA structure of my model here, DNA is structured very similar to a ladder. We have a sugar phosphate backbone that's represented by the sides of the ladder, these black pipe cleaners, and that's where the deoxyribo of the deoxyribonucleic acid comes from, or DNA. So we have our sugar phosphate backbones, and then if you look at these rungs, which there are about 3 billion of those in a single human cell, that's where those nitrogenous bases are. And when we're talking about DNA in a human cell, you're going to find 4 different kinds of nitrogenous bases, which we'll be representing with pony beads today. So we'll start off over here. I have my nitrogenous bases paired together. Guanine is a nitrogenous base, and we're going to be representing it with green beads today. Cytosine is going to be represented with white, and I have them sitting next to each other because they always bind together. They bind together with 3 hydrogen bonds. The other 2 nitrogenous bases are adenine, which will be represented by yellow, and thymine, which will be represented by blue today, and they're always bound together with 2 hydrogen bonds. Now, when we're making our model today, we're going to have to consider the size of the chemical structure of these nitrogenous bases, and guanine, which, if you'll remember, is going to be represented by green pony beads, is a much larger molecule. If you look at its actual structure, it looks like 2 rings compared to the 1 ring of cytosine structure. So to take that into consideration, for every 1 white cytosine bead, we're going to pair that with 2 green beads to represent that larger guanine model. Okay, on this side, same deal. Adenine is a larger structure. It looks like it is structured in 2 rings compared to the 1 of thymine. So for every 1 blue bead, we're going to add 2 yellow beads to represent that adenine base. So let's get started with assembling our model. We have black chenille sticks or pipe cleaners as well as white. The black are representing that sugar phosphate bond or backbone, and the white are going to represent hydrogen bonds and be the rungs of the ladder that those nitrogenous bases are going to sit on. And I'm just cutting these in about 1 to 2 inch pieces, enough space for our bases to sit on. I'm going to create a couple bonds here. So I'm going to add if you'll remember, 2 guanine beads to represent guanine and 1 cytosine. We have some hydrogen bonding going on there, three bonds. And then I'm going to assemble the adenine-thymine bond, which I'm going to represent with 2 yellow beads for adenine and 1 blue for thymine. And we have 2 hydrogen bonds between those nitrogenous bases. And then once you have developed your code and you have lots of nitrogenous bases, you're just going to start attaching them to that sugar phosphate backbone. So I'm just twisting those pipe cleaners on together so they'll hold in place. Like so. And you'll just continue with the code all the way down until you fill in your DNA molecule. So again, you're going to have a sugar phosphate backbone, the sides of the ladder, and then down the center, the rungs of your ladder are those nitrogenous bases. And again, there are 4: guanine, cytosine, thymine, and adenine, represented with pony beads. And once you have completed your ladder, you have to finish twisting it so that your DNA takes that double helix shape. So I'm just going to twist my model and get it looking more like DNA. So thanks for